Dear learners, I'm Dr. Jyoti working as Associate Professor, Department of Botany and PG Studies, Government Science College, Autonomous, Nuputunga Road, Bangalore. I'm going to deliberate on the topic citrus canker, also called as leaf spot. It is also called as bacterial spot of citrus. Citrus plants are native to tropical and subtropical regions of Asia. Citrus plants are believed to be domesticated in Asia. In India, nearly about 10.37 lakh hectares of land was under cultivation of citrus, and nearly 12.053 million tons of citrus was produced during 2016-17. Citrus is affected by many diseases like citrus canker, quick decline, and Phytophthora blight. One of the most important diseases is citrus canker, and it affects almost all the commercial varieties of citrus, and it has caused nearly loss or damage of millions of dollars. Citrus canker is one of the most devastating disease affecting the citrus industry worldwide. Let us discuss about the occurrence of this disease. Citrus canker is a widespread disease, and it occurs in almost all the citrus growing countries of the world. It is reported to have been originated in Southeast Asia, from where it has spread to Europe and USA. The disease is still quite serious in India, China, Japan, and Java. Presently, the citrus canker is occurring in nearly more than 30 countries in Asia, in Indian Ocean and Pacific Islands, South America, and southeastern United States. So here's a pic showing the distribution of the citrus canker disease in different countries of the world. Let us discuss about the symptomatology. The primary symptoms are leaf spotting and fruit blemishing. The most diagnostic symptom is the development of cankers where hyperplasia occurs. Hyperplasia means uncontrolled division of the cell takes place. The lesions they occur on all the parts of the plants, that is, it occurs on leaves, thorns, twigs, older branches, fruits, and in severe cases it is found to even affect the roots. These symptoms they become visible about 7 to 10 days after infection. In case of the leaves, the lesions they first appear as small pinpoint, round, then watery or water-soaked and translucent spots, and these spots may attain a diameter of 2 to 10 millimeters. These are raised, which then later on become yellowish brown in color. They first develop on the lower surface of the leaf and then they spread on to the upper surface of the leaf. Now, as the disease advances, the surface of the spot becomes white or grayish in color, and finally the epidermis ruptures in the center, and this gives a rough and corky and crater-like appearance. So this is a pic which shows the spot that is occurring on the leaf. In the center, you will find the necrotic lesion surrounded by the water-soaked margin, and surrounding that is the yellowish colored region, that is the halo. In this pic you can see the progression of the symptoms of the disease. These spots they then increase in size, and the neighboring spots they may merge with each other, that is they may coalesce to form elongated lesion, especially on the fruits and the twigs. Now these spots are surrounded by yellowish brown to green raised margin and a yellowish halo. The spots also occur on the petioles and the midrib of the leaves, and this causes premature defoliation. Cankers that occur on the fruits, they have got a very prominent crater-like depression in the center. This is this can be seen over here in the pic. In extreme cases, gumming of the spots on the fruits is also seen. Severely infected fruits they drop prematurely, and this leads to the reduction in the yield of the fruits. Though the fruits get infected, still they are edible and they can be used for juice. Thus, defoliation, dieback of the shoots, then fruit drop, and general tree decline are the symptoms of this particular disease. Now let us look into the conditions which favor the spread of this disease. Rainfall and warm temperature that ranges from 20 degrees centigrade to 30 degrees centigrade, and the period when the new shoots are emerging and also during the early fruit development are the conditions which favor the spread of the disease. Now, let us look into the etiology. The causal organism or the pathogen here is a bacteria which is a facultative parasite. Three types of cankers have been identified based on the causal organism. Xanthomonas oxynoporus pathover citri, which is also called now by the name Xanthomonas citri, subspecies citri, causes type A type of the canker. It is also called as Asiatic type canker. This is most widespread and severe. Type B and type C type of canker is caused by Xanthomonas oxynoporus pathover orangifoli, also known as Xanthomonas fuscans, subspecies orangifoli. And type C type of canker is also caused by Xanthomonas fuscans, subspecies orangifoli, type C. This bacteria is gram-negative, it is aerobic, it is rod-shaped, it is capsulated, and it is motile. It is about 1.5 to 2 into 0.5 to 7.5 microns in size, and it is monotrichous. It forms chains and it does not form the spores. There are different pathovars and variants of this bacteria as has already been discussed. The host. All the citrus varieties, that is lemon, orange, grapevine, pimento, mandarin, lime, tangerine, sour orange, and rough lemon, are the hosts for this bacteria. The disease attacks the plants of citrus both in the seedling stage as well as the grown up trees. Let us now look into disease cycle. The infected twigs, which have got the older lesions, they are a main source of infection. Now these lesions, they ooze the bacteria, and this bacteria are blown away by the wind or they may be dispersed by the rain. So here you can see the bacterial ooze occurring on the fruits. Healthy plants are then infected by this bacteria, which then enter through the stomata or the wounds which are caused by citrus leaf minor, that is Phyllonistis citrella, during the feeding activity. So here is a pic showing the bacteria entering the healthy plant through the stomata, and this is of the leaf, a pic of the leaf where the wounds have been caused by the citrus leaf minor. This bacteria, on penetration into the host, it multiplies in the intercellular spaces and then it dissolves the middle lamella and then gets established into the cortex. Now these lesions on healthy plants, they become visible about 7 to 10 days after infection, first on the lower surface of the leaf and later on the upper surface of the leaf. Man is also an important agent of dissemination through infected nursery stock. High mean temperature, which ranges from 20 to 30 degrees centigrade, and the rainfall favors the development of the disease. This is another pic of the disease cycle. Here you can see infected twigs with old lesions. They are the primary source of the inoculum where the oozing of the bacteria takes place. This bacteria are then disseminated by the wind. Then they infect the healthy plants. They enter the healthy plants through the stomata or the wounds. Then it multiplies in the intercellular spaces of the plant. It establishes itself in the cortex and it may spread to the other part of the plant, especially the leaves. So, so this stage represents the dormant stage or wherein the perennation takes place, whereas this is the most active phase. Thus, dissemination of the pathogen takes place through infected leaves with old lesions, then by means of driving rains, then by wind, by insect, by means of agricultural tools, and sometimes even man is the important source through the infected nursery stock. This is another pic of the disease cycle. You can see the citrus plant over here. Once the bacteria it enters the stomata, it invades it invades the healthy leaf through the stomata. Here it will multiply, it will gain entry into the intercellular spaces of the mesophyll tissue, and later on it will dissolve the middle lamella and gain entry inside the cells. In this way, it enters the lower surface of the leaf first and then spreads to the upper surface. So you can see that the bacteria may infect the branches, the fruits, as well as the leaves. Now this, now the bacteria, then it may be disseminated due to the rain. Now this is a pic of how the lesion occurs. So here you can see, when the bacteria gains entry through the stomata, what happens is the lesions they become irrumpent, or a pustule develops. That is because of the hyperplasia. Uncontrolled division of the cells takes place, and that is why the lesions become irrumpent. Now what happens in the center, these cells they then die. When they die, a crater is formed. It is called as necrosis. When necrosis takes place, the cells will die, and therefore a depression or a crater is formed. Whereas surrounding that, over here you can see the water soaked areas. These are the water soaked areas, and besides that you can see the yellow region wherein the chlorosis has taken place, and this actually represents the halo. Now further what happens is, this bacteria it may spread by means of the wind or by means of the rain to the healthy plants. Now other means of transmission is by through the agricultural implements or the tools which are being used for the pruning. When the implements when the implements are used on the diseased plant and such implements are again used on the healthy plants, then the transmission or dissemination of the bacteria will take to the healthy plants. Finally, let us discuss about the control measures, that is the disease management. It can be done by different methods. One, by quarantine measures. Implementation of strict international plant quarantine regulations against the pathogens has to be done, not only at the international level but even uh, from state to state within the country. Cultural control. In cultural control, eradication of the infected trees and then burning them off, then pruning of the infected parts of the plants and then burning the debris off. Use of disease-free nursery stocks should be used for planting in the new orchards, and growing windbreaks on the hedges in order to hinder the spread of the inoculum. Similarly, so also in the orchards, the health of the plant should be maintained by proper fertigation as well as time to time irrigation, so that the vigor of the plant is maintained as optimum. Proper manuring should be done from time to time. The third method is by chemical control. Applying preventive sprays of bactericides, for example, cocides, then spraying of copper oxychloride 0.5% or spraying of Bordeaux mixture 1%, 5 is to 5 is to 50, the ratio of 5 is to 5 is to 50 before planting in the orchards, and thereafter at an interval of 15 days has been reported to be effective as well as economical. Similarly, sprays of antibiotics, streptomycin sulfate, that is 0.05%, then fomomycin and phytomycin 2500 ppm has also been found to be effective. 3 sprays of 50 grams of streptocycline plus 25 grams of copper sulfate in 500 liters of water per acre, one each during the month of October, December, and February has proved to be effective. Fourth method is biological control. Using antagonistic bacteria is being explored. Application of neem cake solution on the foliage has found to be effective. The fifth method is developing transgenic plants. The sixth is use of resistant varieties. SN2, Sankar 3 and Sankar 4 varieties have been found to be resistant to citrus canker. Similarly, use of field resistant citrus cultivars. So in summary, the yield losses range from 5 to 30%. The host is all the citrus varieties. Pathogen are Xanthomonas oxynopolis pathover citri and Xanthomonas oxynopolis pathover orangifoli. This disease affects leaves, twigs, thorns, older branches, fruits, and in severe cases it affects the roots. This disease it disseminates or spreads by wind, rain splashes from the infected part of the plants and also by means of the contaminated agricultural tools. Disease management can be preventive, that is prophylactic, effective quarantine measures by biological control methods and by chemical methods. These are the references that I have used for preparing this topic. Thank you.